Question? Okay. So now, okay. So remember that when we talk about transcription, this is the copy, this is the sequence of the gene. We talked about the promoter and enhancer elements. We talked about +1, and then messenger RNA after plus the polymerase starts copying this DNA into a piece of RNA +1, and polymerase simply just copies until it terminates. It doesn't know the difference of any of the sequences in here. We know, since about 1977 or so, that RNAs have regions that actually are part of the mature RNA, and regions that are not found in the mature RNA that are called introns. Now, these don't always have to be coding regions. In fact, the very first intron was identified in a transfer RNA molecule. It was identified in a transfer RNA molecule. It was a piece of RNA, right, that was present in the primary transcript, but not in the final RNA product. Why do you think it might have been first found in tRNA? I think it might be first found in tRNA. What do you have to be able to do to find it? You have to be able to sequence something, right? You have to sequence the DNA and figure out where the protein coding regions are and so forth. Well, so why would you find an intervening sequence in tRNA first? Well, in 1977, we couldn't sequence our DNA like you did. Right? We couldn't do it. In 1977, we sequenced how big is a tRNA molecule? They vary from 75, 100, and 120 nucleotides, depending on the tRNA it is, but they're small. It took 2 years to sequence them. Right? Not like you guys. You get 6 times that number of nucleotides in one run. Right? One way. It took 2 years to sequence tRNA. So there was no way you could do messenger RNA. No one has ever sequenced a messenger RNA without typical, the new sequencing techniques. So once again, it's the technique and the system that really generate and drive the science. At that time, they could only sequence that small molecule. When they did, they found out that the tRNA in the cell was not collinear with the gene. It had this insertion in there. And of course, they thought they'd screwed up, right? That they'd been sequencing, and somehow they got this piece of tRNA in there, or this piece of RNA in their tRNA, and it was messed up. But it turned out to be an intervening sequence. Messenger RNAs have intervening sequences. They have coding regions. We call them exons. Why do we call them exons? Does that make sense? Why would a coding region be an exon? Well, because we call these introns first, right? Because they were intervening sequences, and we called them introns, so obviously these were ins, these had to be x's. So exons are coding regions. That's the only reason to call that. When you look at the transcript of this gene, what RNA polymerase makes, that transcript is actually exactly collinear with the gene itself. It has untranslated regions, or untranslated region, right? It's transcribed, but untranslated. Usually in the 5' end, it's about on average is 100 nucleotides or so. And then there's exons and introns. The number of exons and introns can vary tremendously. And then at the end, at the 3' end, there's an untranslated region, and that's often big. It can be 600 nucleotides. And then there's a termination site. Okay. Product of a mature RNA that leaves the nucleus and gets out of the cytosol doesn't have introns. It has a 5' cap. This is actually not very good because they should have a 5' cap here, right? I just told you that as this is being transcribed, it gets a 5' cap. So this has a 5' cap. So does this guy. It then has all the exons put together so they're linear. There's no spaces, there's no introns. Reason being, ribosomes cannot tell the difference between an exon and an intron. They only read triplets. If you had this, they would start reading here, and they would just go through and they would read this as triplets encoding for protein. So in order to make a mature protein that's the correct protein, you have to have these exons put together. Okay, and then at the end, the poly A tail is put on. The number of introns varies tremendously. You can see that in the globin gene has three exons. This happens to be one for factor VIII, one of the clotting factors, and it has 25 exons there. The other thing that you can see They're about the same size. They're pretty much, the exons are about the same size, and they're about, they're 100 or so amino acids. They code for 100 amino acids. But the introns, which are the yellow regions here, vary tremendously. They can be as small as they are here, with the 2 introns in beta globin, or they can be very large. Some of these things, the exons can be 5,000 base pairs long. Not unusual at all. Some of them even longer. Because of that, right, the primary transcripts can be extremely long. And in fact, if you look at the primary transcript of an messenger RNA, right, so this is the primary transcript, and this is the messenger RNA. Primary transcripts that contain the exons are on average 10 times as large as the messenger RNAs, even though introns and exons are 1 to 1. There's about 10 times as much exons or intron as there is exon. The number of these can vary tremendously. A lot of the proteins that serve as extracellular matrix proteins and stuff like collagen are very, very large. The collagen gene has 50 exons in it. So the exon, there's exons of 50, average 50 to 20, 250, so 100, 150 amino acids is average. And no, that's not exon. I should say that introns are 5 kilobases. I guess this is the total exons are about 5 kilobases. That must be what I was writing. But the exons are 45 kilobases. So if you take the collagen gene and transcribe it, it's 45 kilobases long. If you remove all of the introns, it's only 5 kilobases long. So it's about 10-fold difference. So it's the same thing I said. Okay. So the question is, how are these guys removed? Do we have to remove these introns? How are they removed? Well, we guessed very early on that there had to be something in the mechanism that was very similar or that was repetitive, that was used over and over again. Why is that true? Why do introns have to be cut out of RNA, primary transcripts, in a very common, in a similar method? Why can't they all be cut out separately? It's really, I guess it's kind of a dumb question. We have 20,000 genes. All 20,000 of those genes, right, are coding for RNAs that have multiple coding regions that are spaced there. Because if you have that, you have to have 200,000 genes to code for the proteins to take them out in an individual fashion. So it had to be very uniform. And it is. If you look at this, so here's an exon in blue and an exon 2 in blue. This is the intron in between, and there's some very common features. There are these consensus sequences, and if you look at the 5' side of an intron, this consensus sequence is conserved. Remember, it's a consensus, it's not identical. The only thing that's perfectly conserved is the GU. So the GU on the 5' side of the intron is perfectly conserved. On the 3' side, there's also a conserved region. What are Y? What's Y? Pyrimidines, right? What's N? Any nucleotide, any nucleotide, what's a U? Purine, so either. So this could either be, this could be a C or a T, this could be so forth, whatever. But this is conserved, but the AG is perfectly conserved. So on each side, the 2 nucleotides that abut the exons are perfectly conserved. There's a third conserved site. It's called the branch point A site. It has several nucleotides, but that A is perfectly conserved. Branch point A is perfectly conserved. This particular guy occurs average about 30 nucleotides upstream from the 5' end, or the 3' end of the intron. Don't forget, this intron could be 15,000 nucleotides. There's a break there. So this is very close to this 3' end, not to the 5' end. And in processing, what we have to do is that intron has to be cut out, it has to be removed, and the 2 pieces of RNA have to be put back together, which, of course, defines a new molecule. In order to do that, what do we need? An RNA ligase. Until people found that tRNA intron, no one had ever found an RNA ligase. But we do have to put have an RNA ligase to put those back together. Why? when you cleave this guy here, right, when you make a cleavage there, what's on the end of that nucleotide? What's on that G?
The
sugar? No, the sugar's not on the end. What's on the very end? Phosphate, right? This is a sugar phosphate backbone again. So it's a phosphate on the end of that. And what's on the end of this guy here? Hydroxyl group. What's on the end of the 5' end of this exon?
Base.
A monophosphate, and then G has a 3' hydroxyl. So to put that 3' hydroxyl and that monophosphate together, there is no energy there to do the biochemistry. Right? So what we need is a ligase molecule to put it back together. Right? Okay. So how do these guys cut out? How are they recognized? What's the function of these guys here? Well, so what happens, first of all, is it shows you here that this is the intron. And the intron is cut out in the structure of the hollow area because it looks like a last neuronal area. And essentially what happens is that this branch point A there, the hydroxyl in the branch point A, it undertakes a nucleophilic attack on that linkage right there. The linkage between the G and the two G's in this case. So you can see that branch point A undertakes a nucleophilic attack on that region. Why does it do it right there? Why does it do it between those two? What does that say we have to have? Remember, these introns can vary in size from a couple hundred nucleotides to 50,000 nucleotides. This branch point A is always, 30 or so, average of 30 or so from the end there. So how does it always find the right point there? What that says is there has to be a mechanism to locate and identify, right, that end of the intron. There has to be something that should happen. But what happens is it goes like this, right? And you have this nucleophilic attack. And you end up forming this region right here. This A is very, very different now. Why is it different? Why is it different? So here's this branch point A, and there's the hydroxyl. Which hydroxyl is that?
2'.
It's not the 3' hydroxyl, because the 3' hydroxyl is divided in the phosphoester linkage between it and that pyrimidine there. So it has to be the 2' hydroxyl. So it's the 2' hydroxyl that undertakes that nucleophilic attack here and forms this linkage here. So now what we have is a phosphoester bond between the 2' hydroxyl and the phosphate of that A. So now why is this A different? That's the only nucleotide that I know of, and there may be another, but that's the only one I know of that has 3 phosphoester bonds, right? The phosphoester bond here, the phosphoester bond there, and the phosphoester bond here. So there's a triple phosphoester bond in the area. Okay? Then what happens now, when this guy's cut off, right, there's a 3', this guy here, there's a 3' hydroxyl. That hydroxyl can take a nucleophilic attack here. And cause that cleavage there. Okay, we'll keep going on this